So we just talked about monosaccharides. Now we're going to talk about polysaccharides. So polysaccharides are complex carbohydrates, are polymers of monosaccharide monomers. So that was a mouthful, but basically monomers come together to make a polysaccharide. In two sugars, just two of those monosaccharides linked together is called a disaccharide, like maltose. Sugars are linked together, like all the other macromolecules we've talked about, in what's called a condensation reaction, right? So water comes out. And this forms what's called a glycosidic linkage. And we're going to talk a little bit about this, okay? And it occurs between two of those hydroxyl groups that stick off from a ring. And the linkages are then broken apart in hydrolysis reactions, right? So you can break apart starch into disaccharide maltose, right? And that happens via a condensation reaction. So that means that water has to go in. Now water's on this side of the reaction, and then it breaks up into your monomers or your disaccharides. Breaks up that glycosidic linkage. So glycolytic linkages can form between any two hydroxyl groups, okay? So remember in the last video we talked about in individual monomers of glucose, if the hydroxyl groups are pointing down, then it's the alpha form, okay? And when those two hydroxyl groups combine, right, they form maltose. And the name of that glycosidic linkage is called an alpha for the type of glucose it was, and then 1,4, so the carbons of those rings that got joined together. So it's an alpha 1,4 glycosidic linkage. This is an example of a beta glycosidic linkage. So here in beta galactose, it's pointing, the hydroxyl group's pointing up, and in a condensation reaction, right, so they're going to come together. This is water coming out, then they're going to be joined together. But the name of the linkage is a beta, right, because that's the monomer that was joined together, and still the number of the carbons, a beta 1,4 glycosidic linkage. And there's different, again, because of this, there's different geometry to those bonds. So now I'm going to talk about polysaccharides. And what I want you to do for all the polysaccharides is tell me the name, the monomer it's made of, the types of glycosidic linkages found in that polysaccharide, and if there's multiple strands, how are those strands linked together, and then what is the function of that polysaccharide. So first we're going to talk about starch. Here's bread made of starch. And the monomer in that polysaccharide is alpha glucose. And alpha glucose is linked to another alpha glucose via these alpha 1,4 glycosidic linkages, right? So just the ones we've been talking a lot about. And if all that's present is alpha 1,4 glycosidic linkages, that starch molecule is called amylose. And if they're alpha 1,4, but every once in a while you get an alpha 1,6, kind of creating a branch, that's called amylopectin. And so here's an example, right? One long strand is called amylose, and then when it's branched, because of this alpha 1,6, every 1 in 30 monomers, you get amylopectin. And there's no parallel strands, and the function is plant sugar storage and plant energy. So cells are going to make these polysaccharides and use them to store sugar and then create energy when they need it. So the next one we're going to talk about is glycogen. Okay, so here's an example of muscle where animals store their glycogen. And the monomer is alpha glucose. And the linkages are alpha 1,4 glycosidic linkages. And now every 1 in 10, so a lot more frequently, we have this alpha 1,6. And so we end up with a lot more of a branched polysaccharide. There's no parallel strand linkage, and we use it for energy storage in animals. And this energy is stored in our liver and our muscles. And these are places where we need a lot of energy. So as you know from lab, right, we have these polysaccharides, and enzymes are what's responsible for hydrolyzing those polysaccharides down to simple sugars so that the cells can use it. And so starch and glycogen are easily hydrolyzed because they have these alpha glycosidic linkages, and we have the enzymes to break those apart. Starch is hydrolyzed by an enzyme called amylase, as you know, and glycogen is hydrolyzed by the enzyme phosphorylase. So we're able to break them up, and then we're able to use the glucose. The next thing I want to talk about is cellulose. This is a carbohydrate that's found in plant cells, and it's made up by the monomer beta glucose. Okay, so that's different than the previous two. And because it has beta glucose, we end up with these beta 1,4 glycosidic linkages. And we don't have the enzymes to break apart cellulose. And so when we talk about cellulose, we talk about dietary fiber. So this isn't something that our body can easily digest. And okay, so we have these beta 1,4 linkages that create a strand, and then strands are linked to one another by hydrogen bonding, right? So that electrical interaction between partial charges on hydrogen and other partial charges. And so we end up with this essentially fiber, right, of multiple strands binding together to create a very strong structure. And so the function, right, kind of relates to its structure in that it forms the cell walls of plants. So a very stiff, rigid structure that allows plants to stand up. The next thing we're going to talk about is chitin. Okay, so I have Sebastian down here because he's a crustacean. And the monomer that makes up chitin is called N-acetylglucosamine, or NAG. And it has these beta 1,4 glycosidic linkages, and every other monomer is flipped over. And they make these strands, and then those strands hydrogen bond with each other. So the difference in this case is the monomer. And so here's just an example of these NAG monomers, and then they interact with each other via these hydrogen bonds and create this stiff structure that that makes up a cell wall in fungus and the exoskeleton in insects and crustaceans. So a really tough structure. And the last one, and we've talked a little bit about this already, is the polysaccharide peptidoglycan. And so peptidoglycan is made up of repeating units of NAG and NAM. And they're linked together via these beta 1,4 glycosidic linkages, beta. And their parallel strands are actually linked together by peptide bonds that form between short amino acid chains. And so here's an example. So you have NAG, NAM, right, repeating. And then here these bonds, instead of being hydrogen bonds, are actually covalent, right, peptide bonds between the amino acids. So it's a very tough interaction between the parallel strands. And it functions as a bacterial cell wall. So what do carbohydrates do? Carbohydrates have diverse functions in cells. They serve as precursors to larger molecules. We've talked about this already in the case of nucleotides, but also in amino acids. They provide fibrous structural materials, so peptidoglycan, cellulose, chitin, and they also indicate cell identity, and they store chemical energy. So we're going to talk about these last two. So cell identity. Carbohydrates indicate cell identity, and carbohydrates can modify proteins and lipids. So we talked already about proteins, right, made up of amino acids, and we'll talk about lipids later. But little carbohydrates or oligosaccharides can be added to these lipids and proteins, and that helps another cell recognize itself, right? Identifies cells as self, or it helps communicate between the two cells. So an example of this is blood types. So Carl Landensteiner's research was recognized for discovering the blood types and mapping them out, and he won the Nobel Prize in 1930. But basically what the cell types are is that, let's take the O blood type, for example. You have a lipid, right? So this is a glycolipid, and it's had these sugars added to it. So glu, gal, nag, gal, and fructose. Now, if you have the A blood type, you have an additional monomer added at the end, this gal-nac, okay? And that gives you the A blood type. And if you have the B blood type, you have this base, but then you have a gal at the end. And the reason this is important is that our immune cell, if we have an O blood type, we recognize A and B blood types as foreign, and our immune system rejects those blood types, right? Whereas if we donate O to this A and B, it doesn't have any extra modifications. So if you're an O blood type, you can donate your blood to anybody. You're a universal donor, right? And if you have the AB blood type, so you that means you can get blood from anybody else. You're the universal recipient because you have antigens for both A and B. And so there's another example of cell identity in the book, and that's Wassermann's experiments, and so you should read about that. So the second thing I'm going to talk about is storing chemical energy. So in photosynthesis, plants harvest chemical energy from the sunlight, so energy from the sun, and they store it in bonds of carbohydrates. So this is a complex process we'll talk about later, but the basic reaction is that CO2, water, and sunlight equals sugar and oxygen. And in cellular respiration, cells can metabolize or break up the bonds of carbohydrates to release energy. So how does it do this? How does it store energy in chemical bonds? So let's talk about this a little bit. So nonpolar bonds are sources of potential energy in cells, okay? So here's an example of a polar bond, okay? And so we have water, so H2O, and we have CO2. Now these are polar covalent bonds. So that means that oxygen holds those electrons more tightly than the hydrogens. And in CO2, oxygen holds the electrons more tightly than the carbon. And so because of those polar bonds, because of those tight chemical, or the tight holding of electrons, those molecules are kind of more stable. And so because of that, they have less potential energy. So I have an example of that here. We have oxygen and a carbon and hydrogen, right? And this is oxygen, and it's going to hold the electrons more tightly when it binds to carbon and hydrogen. It's going to make a really tough bond. And so it doesn't hold a lot of chemical potential energy. Okay? Glucose, right, has a lot of carbon and hydrogen bonds, okay? And carbon bonds, okay? And so carbon-carbon bonds, the electrons are shared evenly, and carbon-hydrogen bonds, the electrons are shared evenly. And so these bonds are easier to break. And so these bonds can come apart pretty easily, and then if you then make covalent bonds instead, that potential energy in those bonds is released, right, to form the more stable bonds. And so if you think about carbon and hydrogen, right, we had carbon and oxygen held together by magnets. You had to pull them apart. They were really stable, had very little potential energy. Carbon and hydrogen, right, these bonds are weak, and they just pull right apart, right? But they store lots of energy. So since energy is never created or destroyed, metabolizing or breaking up nonpolar bonds and then making polar bonds releases energy. And so if you take cellular metabolism, we take glucose and oxygen, a molecule that has lots of carbon-carbon and lots of carbon-hydrogen bonds, and we break those bonds and we form molecules that have polar covalent bonds, so CO2 and H2O. If we do that, we release the energy in that. And we actually release that energy in a way, and we store it as ATP. So here's a molecule of ATP. You have your ribose and your base, and then you have these 3 phosphate groups. And these phosphate groups have all these negative charges on them. And so those negative charges are a really good way to store that chemical energy from glucose. And then we can use that ATP then to power more reactions. And we'll talk about that when we talk about energy. So here's carbon dioxide, right? There's very little potential energy in this molecule because of those polar covalent bonds. Carbohydrates we just talked about. Lots of nonpolar bonds, so lots of energy, potential energy in that molecule. And then fat, right? Here's an example of a fatty acid, has a ton of carbon-hydrogen bonds. And so fat is a really good source of energy. So to summarize, specific enzymes are required to hydrolyze those carbohydrates. And structure, as we talked about for all those polysaccharides, gives rise to function. So building blocks, support, cell identity, and energy. And that's all I have to say, and I look forward to seeing everyone in class. Thanks, bye.